Good morning, good afternoon, and good evening, everyone. This is Liam Sanyo from Inside Scientific, and I'm very pleased to be your host for today's event, which is titled Hippocampal Neural Circuits in Awake Head-Fixed Mice Navigating a Real-World Environment. Today, we're fortunate to be joined by Professor Simon Schultz, Professor of Neurotechnology and Head of the Neurocoding and Neurodegenerative Disease Lab at Imperial College London, and Professor Michael Goard, Associate Professor in the Department of Molecular, Cellular, and Developmental Biology at the University of California, Santa Barbara. Today, they're going to be discussing their research using two-photon microscopy to study the neural circuits of the hippocampus in awake head-fixed mice navigating a real-world environment. Okay, now without any further ado, I'm very pleased to introduce our first speaker, Professor Simon Schultz. Simon, thanks so much for joining us today, and feel free to take it away whenever you're ready.
Okay, so today I'm going to tell you about some recent work that we've been doing, looking at place cells in head-fixed mice navigating a floating real-world environment. So this work has been done at Imperial College London. You can see the lab that it's been done in is sort of somewhere back in there, sort of behind the sort of front entrance of the Royal School of Mines building in London. Now, our motivation in coming to this work was really came out of a line of research on understanding the neural circuit basis of neurodegenerative disorders. So dementia is, as I'm sure you're aware, a growing problem for the world. Dementia sort of results in deterioration of memory, sort of thinking and behavior, and is affecting a large number of people worldwide. So around 50 million people worldwide have dementia, and of these people, Alzheimer's disease accounts for 60 to 70% of these. Now, if we want to make progress on this problem, we really need to understand the basic neural circuit mechanisms that underpin the deficits that give rise to the problems in dementia. If we can do that, then maybe we can start to use that to sort of steer the development of new treatments. Now, in so doing, mouse models of various aspects of neurodegenerative disease are extremely important. And I would argue that in order to make best use of these, we really need a technology platform which lets us get at both behavioral issues that are stemming from dementia, at changes in neural circuit structure and function, but during cognitive and memory tasks which are affected by dementia. So this project was really sort of originating from the need to develop a technology platform, or bring together a technology platform, if you like, to get at these kind of questions. So to do so, there are a number of technology approaches we could make use of. And there were really sort of three technology games in town at the moment, I think, that sort of lend themselves to this. One of them is high-density multi-electrode arrays. So most recently illustrated by the NeuroPixels array, for instance, which really give us unprecedented sort of levels of ability to record from large sort of densely packed sets of cells in the brains of behaving animals. So that one approach, because the trouble is we're just looking at an electrical signal there. We don't actually sort of see the cells. We can't exactly localize where they are, and we're sort of restricted in finding out further things about them. Nevertheless, it's a powerful approach which will be very useful. Another approach which has actually been developed quite a bit recently is the miniscope, where you have a head-fixed microscope which is attached to the head of a mouse or a rat. And that allows you to do single-photon fluorescence imaging, where you might have a genetically encodable calcium indicator, for instance, in the mouse brain, and you can do wide field imaging and pull out the activity of sort of many, the signals of many hundreds of neurons. Now, the trouble is that the optical signals you get are not sort of separated in the Z direction in depth, so you need to demix those. And we are getting better at doing that, but nevertheless, the imaging that you get out is not as good as two-photon imaging. So it's this technology on the right, multi-photon imaging, we'll be talking about here. And as you can see from the rotating maximum intensity projection there, taken from the layer 5 pyramidal cells in the cortex of a mouse brain, it gives you sort of exquisite spatial resolution and very good temporal resolution on cells, but of course it requires head fixation, and that has a lot of limitations associated with it. So how can we do behavior under head fixation? How can we do interesting behavior under head fixation? And we want to do memory tasks, so actually that's maybe particularly challenging. So we wanted to image neural activity during a memory task. So here's the general sort of setup that we use. So over on the top left, you can see basically a sort of schematic of our apparatus with sort of a mouse brain. Now we actually have a window into the brain. We aspirate a small area of cortex above the hippocampus. So we're particularly interested in looking at the hippocampus, and in particular, we're going to look at our area CA1 of the hippocampus, wherein you find the famous place cells. And so we aspirate a small area of cortex above it down to the level of the corpus callosum. We sort of make a cranial window there by putting a small cover slip in. And you can see we have the objective lens of the microscope effectively sitting directly above the brain of this mouse. Now we have to label the cells in some way as well. And we, in this project, we've used GCAMP6, the genetically encodable calcium indicator. We use GCAMP6S, which has got sort of quite good sort of signal to noise ratio, quite good signal amplitudes obtainable from it. And in what we did in this project is we actually used a construct which gave it together with MRuby. So we used GCAMP6 as a green indicator for calcium, but in the cells where we put it in, we also transfected with MRuby, and MRuby just gave us a sort of a fixed sort of static marker of the cells. So you can see in this image, the top middle here, there's both a sort of an overlaid green and a red channel. You can see sort of red basically corresponding to sort of the interior of cells, and sort of green around the edges, and a few cells you'd happen to see just green there. Now, the reason for using a red marker as well is because we actually want to track these cells over multiple days. Then, the nice thing about GCAMP6S compared to earlier calcium biosensors is the signal amplitude was increased, and that's gone on. We now have sort of GCAMP7, etc. But they did that basically by reducing the baseline fluorescence. And so that means if you want a sort of a stable signature to track over time, it's not really the green channel is not the best one to choose for that, because of course which cells are active at any given point in time is going to change. So it's very useful to have a red static marker as well. And so in our case, we've used this MRuby. It could be something else. It doesn't have to be delivered together, or you could have a separate virus to induce red fluorescent proteins in some cells. You could also be using that to label particular cell classes as well, and so kill two birds with one stone in that way. So that sort of lets us look at the activity of an area of cells here. And I should say this work is being done primarily by Dr. Marianne Go, a postdoctoral fellow. You can see a picture on the right here, together with Jake Rogers, who was another postdoc who specialized in the behavioral side, and Katie Davey and Siegfried Prado were involved in the analysis work here. So you can see over on the bottom left there, the sort of flashes of sort of calcium from populations of cells in actually 3 times actual speed. So you can see the sort of the patterns of coming up there as the cells are active. Now, the additional thing we can do with this is to co-label cells that are, or sort of co-label areas of tissue which are affected by amyloid plaques. And we've done that using methoxyXO4, and we basically inject that intraperitoneally. We move the laser excitation wavelength down to 720 nanometers, and basically the first few days of sort of imaging, we actually spend sort of mapping the brain, finding out where the cells are, and also seeing where the cells are relative to the plaques. The plaques are sort of shown here in this sort of purple channel. Okay, so that's what we're looking at. Now, how do we do the behavior? So this is the apparatus. We have head-fixed mice running in an air-levitated cage. So that's the this sort of Neurotar mobile home cage. You can see down here, there are basically sort of little air jets which sort of result in this carbon fiber cage floating on a bed of air. The mouse is head-fixed, and when the mouse is moving, for instance, it's actually the cage that moves rather than the mouse. And this is all under the two-photon microscope there. So this sort of shows the apparatus in action. You can see an example here of a mouse running in a circular track. So this is the most common track we use, where it's just sort of running around and around in a circle. But you can put sort of various other tracks as well. And you can see here another example with a circular track with a stop and a Y maze and so forth. And we've done the same thing with a fully sort of open field environment. One thing that's important to note is that in this apparatus, and actually I'll just sort of go back and sort of show the apparatus again. You can see, you're seeing this here in the light, but of course normally when we're doing these experiments, it's in the dark. Now, we actually intentionally don't illuminate the background, and that's because if there were distal background visual cues, they would be, so the animal's fixed in one place, so they wouldn't be moving with the animal, if you like. So there'd be a confounding stimulus. So we're doing this in the dark, but we do have proximal visual cues on the maze itself, and these are actually phosphorescent tapes. So basically, at the start of each behavioral session, we effectively charge them up by switching the light on, and then they sort of slowly decay over sort of tens of minutes following that. And these result in visual cues that the mouse can see, the scotopic rather than photopic vision, so sort of dark adapted vision, and there are also tactile cues as well. So the mouse has proximal visual and tactile cues with which to recognize where it is in the environment. And mice learn to do this over several weeks. Actually, sort of typically a week or so is sufficient in our experiments these days. And you can see 2 examples here. One where at the top, where you've got basically the mouse running around a circular track, and the bottom is an open field environment. And you can see over sort of 14 or so behavioral sessions, of which some of those are sort of multiple sessions per day, they learn to run. We measure basically the performance on this task effectively by what fraction of the session that they're actually running for. And so they end up basically learning to run for a water reward a substantial fraction of the time, which is what we want in order to map place fields and basically run our memory task in effect. And similar in the open field environment to the circular track. Now we can ask questions about memory in several ways with this. The first of them is that so now over the week or so in which we do our behavioral training, they're typically exposed to two environments, which we call FAM1 and FAM2, familiar 1 and familiar 2. So these environments they know very well. Now, at the start of each imaging session, they'll typically spend 20 minutes in FAM1, which we consider our reference environment. If we then put the animal into FAM2, we basically just sort of switch the cage on the platform there. What you expect to see is place field remapping. Okay, effectively, the set of place fields in the hippocampus, the cells responding particular locations in place, constitute the spatial memory in effect. When you move it into a different environment, effectively it remembers it's a different spatial environment, and the place fields remap into and we'll see later on how that actually works in practice. But we expect them to sort of remap corresponding to its spatial map of this new environment. So that's like memory recall. It's spatial memory recall. On the other hand, if it spends 20 minutes in environment FAM1 and then we put into a novel environment which it's never seen before, then that's a sort of like a memory encoding task. It's learning, and we could be imaging, during this time, it's sort of learning this new environment, laying down the new spatial memory. And we image at the same time as this. So you can see a typical example here. We've got a 300 micron sort of square field of view. We typically now do them with 500 micron square field of view. And you can see the green channel, the red channel, and then on the right here, there is basically the ROIs, regions of interest, corresponding to a relatively large number of cells that we've extracted, in this case using the Cayman platform. So we have effectively we have our own platform, but it uses our own pipeline, which uses as part of it the Cayman pipeline for extracting individual cells. Now, having done that, we can then pull out the calcium time series corresponding to each of these cells. So just to explain what you're looking at here, so the first row here, shown in magenta, is actually the spatial position of the mouse. So the circumference of this circular environment is 100 centimeters, and it's basically running from running continuously around it, and you can see its position sort of advancing through there. Now, as we're doing that, we can basically extract the time series from many cells. We're just showing a small subset of them from here. And we've got a continuous time series there. We then extract events, and we keep both the time and the amplitude of each of these calcium transient events, and that's what you're seeing down below here, plotted with so there's a dot for every event, and the radius of the dot corresponds to the amplitude of the event. So it's sort of like a spike train, if you like, but there's a bit more information because we're keeping the amplitude of each of these events. And it's worth keeping in mind that these calcium transients may correspond to multiple action potentials. We're not trying to sort of resolve the action potential time series as a spike train per se. We think that's too hard for cells that you don't have a simultaneous recording from in order to sort of calibrate the calcium to spiking relationship. But we can basically keep this as a sort of measure of activity. Okay. And what do we see? So in the circular track, you can see here there's sort of 5 examples, very typical place cells here, each of which fires basically when the animal is in a particular location around the track. And you can see these examples are sort of fairly reliable. They tend not to fire anywhere else. They tend only to fire in this one part of the track. We can, for each lap around the track, we can sort of bring that out into a matrix where we've got position along the track on the x-axis, the lap number on the y-axis, and we can get a sort of a raster like thing here. And you can see here there's a sort of fairly systematic relationship. The cells are really responding at particular locations sort of systematically there. So we have place tuning. Okay. We can average across all of those laps and then order the cells by the location, their preferred location for firing, and that's what you can see on this plot over on the left here. Pulling all of those together, we can sort of pull all of the cells, in fact, across multiple animals, and we'll see something like this next plot here, which sort of really shows this sort of nice diagonal line corresponding to a fairly uniform sort of distribution of tuning preferences. If you actually look into that a little bit more deeply, the next figure along here, and you look at where these place field preferences are, there is actually a little bit more information there. So the cues, the visuotactile cues, tend to be in particular they are in particular locations on this around this circle, and then in addition there are two doors which are used to introduce the mouse sort of into or out of the maze. And it seems that you have more place fields at those door locations, the entrance and exit locations. The place fields tend to be between rather than on top of the cues. And they tend to be in this environment about sort of 20 centimeters or so in length. Okay. Their spatial information content is actually quite high. It's sort of around 1 bit per event there, which is sort of comparable to what you see in sort of freely moving or tethered animals. Now, one thing we were interested in looking at was comparing the cells like the ones I've just shown you, which had place field tuning, to some of the other cells. So obviously there are many cells that we might record which don't have place tuning. And how do these differ? The first thing we can do is we can just look at the event rate, like the rate at which these calcium transients are occurring, and they seem to occur at a higher rate for place cells than for non-place cells. We can actually do a slightly more nuanced analysis where we, over a particular time, window of time, we can add up the amplitudes of these events and then divide by the time window, and then we've got a sort of an activity measure in delta F on Fs per second, and that too, shows that same trend, that the place cells tend to be more active than the non-place cells, which is not particularly surprising. Now, what about the variability? That was something we're interested in getting at. Each time the animal goes around the track, and you can see over on the top left a little sort of circle with a little box there. So each time the animal is passing through a particular location, we can sort of add its activity to a list, if you like, and then we increase that list as we accumulate more and more passes through that box, and then we can look at the mean of that activity over the passes through the box and the variance of it. Now, generally with neural activity, you expect that the variance increases with the mean. In a Poisson process, the variance will increase proportionally to the mean, but in general, it's going to be proportional to the mean to some kind of power. And that power, beta here, we can think of as sort of as a variability exponent. This is an analysis that's been done more commonly in visual neuroscience literature, and we sort of brought it into the place field literature here. So you can take two examples, for instance, and plot the variance against the mean for different locations of that box. So you can change the mean by moving that box around the circle. Some areas got, like in the place field, a higher firing rate, etc. And that gives you sort of many points. You can fit a line to that, and then the slope of that gives you that variability exponent. So there are two examples shown there. And then on the right, we're sort of showing the entire population here. And the interesting thing you see there is that actually the place cells have a lower variability exponent, so they're actually more reliable for the same, for a given firing rate. They fire more reliably than the non-place cells. And we just sort of note this because we really think this might be an important signature of circuit changes in dementia. There's a major hypothesis there, the aberrant excitability hypothesis, that this might be an interesting way to look at that, or other changes, of course. Okay, now that was this sort of circular track. What about the open field? the short answer is we see good place fields in the open field too. So we can see 4 examples there. One thing to note, and you can see that we're sort of showing this on day 1 and day 3 there, they tend not to be stable over long periods across many days. This may be something that's slightly different in this sort of floating environment, where, remember, we've got the animal's head fixed, so the vestibular information is not as you would expect in a normal animal, and maybe that's something that sort of helps stabilize these over longer periods of time. But again, the information rates are actually quite high and comparable to what we see in sort of freely moving animals. These place cells remap across environments. So if you take the animal from one environment into another, as I said, we expect to see place field remapping, and so we can see that if you look at this figure on the lower left there, you have basically the set of place fields sorted according to one environment. If you then put the animal in environment B and use that same sorting, it just looks random because there's there is maybe some tuning in that other environment, but it's the sorting from but basically that because they've remapped, you can't then see this nice straight line anymore. If instead you sort those same cells on the basis of their place field in environment B, you can see the image like on the right, and you suddenly see this nice diagonal line again. So we have remapping across environments. Where do the cells remap to? Basically, the short answer is anywhere. It doesn't seem to make any difference. It's where a place cell had its preference in one environment, you put it into the other environment around this circular track, and it just seems to sort of randomly locate. Okay, that was remapping across 2 different environments. But what about if you take the animal from one environment on one day, and then you go and put it back on the following day into the same environment? And so we're looking sort of over longer periods of time. And the place fields do shift dynamically over time. You can see some examples on, say, cell 143 on the left there, which actually is relatively sort of stable. You can see the next one down, though, seems to be shifting between sort of days 3 and 4 there. So we're shifting its place field. The one below that seems to have sort of shifted and then moved back maybe. And the one below that just sort of shifted completely. So there is a sort of substantial sort of shift over time, even in the 1D environment. And again, this, if you look at sort of neighboring days, they're reasonably close, but then the shift is sort of becoming further and further. And, over sort of 3 or 4 days, you have a substantial shift in the place fields. Now, in freely moving mice, at least from one major study on that, there seems to be more, a longer duration stability over time. However, in virtual reality environments, it's actually very similar to what we see here. So it could be this is a feature of the head fixation, or it could be something else. Now, finally, what we're able to do was to look at not just single cell properties, but the properties of the entire ensemble of cells. And the way we do that here is we take the data, we put it into a matrix of cells by time, okay? And at each, we can say take 2 points in time. At each point in time, we can we extract a vector, which is basically the pattern of activity over all of the cells, and we can compare that pattern of activity with the pattern of activity at a different point in time. Now, so those are vectors, so we can have the angle between them, and the cos of that angle is what we think of as the cosine distance. So we then make this time by time matrix, where each element in the matrix is basically the cosine distance between the patterns you see at those points in time. And having done that, you can then do dimensionality reduction on that matrix, and we do that with multidimensional scaling. As you sort of see, it works with a number of different methods. And you can we're looking at an example in 3 dimensions there. And we can study how that works as a function of how many dimensions we include there. And, we find that, as you sort of increase the dimensions, you can explain more and more of the variance. And we found that compared to at least sort of two other methods, principal components and analysis and Laplacian eigenmaps, that the MDS method we were using in the variance explained measure was slightly better. Now, what does this look like for our circular track? We can see that in 3 dimensions there. We can even just look at this sort of 2-dimensional version on the top right, and you can see that in 2 or 3 dimensions, you're really capturing the structure of the environment that the mouse is exploring. So you can see sort of the color code really shows, like, as the mouse is going around the environment, the pattern of activity in the brain is sort of systematically changing to reflect that. Slightly more complicated in the open field environment, but in part because we've got 2 variables here. Instead of just sort of one environment, the angle around the track, we've got the angular position and the radial position. And the angular position seems to be a little more sort of salt and pepper, sort of less structure there. There is some structure there for the radial position. And we can basically just sort of color code those separately and sort of look at the structure of the representations there. If we compare these, the circular track and the open field, we can look at the amount of variance explained as a function of the number of dimensions of the manifold we're looking at, and then we can sort of count up how many of these dimensions or eigenvalues do we need to capture 90% of the variance. We can call that the dimensionality of the manifold. And then looking across on the middle plot here, you can study that dimensionality against the number of cells that we're including in our analysis, and you can see the dimensionality of the open field is sort of systematically much higher. So the open field representation is systematically much higher than the representation of the circular track. Now we can actually then sort of look at that by trying to decode the variables. In this case, we're decoding the angular position, and maybe unsurprisingly, we're able to do that much better for the circular track. Okay, I'm going to skip that one just to say that it doesn't really matter much what method you use, what manifold learning method you use. But the final thing I'd like to talk about is just sort of a brief taster that you can, of course, use other tasks with this as well. And the one thing that we've done is to look at a sort of a simple spatial working memory task called forced alternation. So the mice sort of run around. We've called it a Y maze, but really it's a sort of a theta maze here. They go one way on one pass, and then the next time they have to remember that they went that way and then go the other way instead. So they learn this very well over several weeks as well. So we're now able to sort of use this environment to ask questions about different memory tasks as well, like spatial working memory tasks. So finally, I'd like to finish by sort of summarizing. We've used the floating cage system to map place cells in 1D and 2D, and we've sort of really validated the system as a method for doing that. We've been able to image calcium signals in populations of neurons during cognitive tasks. We've applied that to spatial memory, the circular linear track and the open field task. We've shown that working memory tasks are also possible with this platform. We're able to observe the recall of old and the formation of new spatial memories, and we're able to track the same neurons over several weeks. We find high spatial information content, which is relatively similar to in range to that you find in freely moving mice or tethered mice. The one thing we would note is that the remapping of the representation of the same environment on different days seems surprisingly fast. I mean, you expect this to occur, but it occurred over a slightly faster timescale than we had predicted. So thank you for your attention. I would just sort of finally like to end by acknowledging some of the people involved, in particular Marianne Go, a postdoctoral fellow who led on this work, Dr. Yu Liu, the lab manager in the group who helped with the work as well, Siegfried Prado, who helped with some of the analysis work, Jake Rogers, who helped a lot with the behavioral work, and then finally on the spatial working memory task, Yimei Li and Gina Song, who were involved in sort of getting that working memory task up and running. So thank you for your attention.
Excellent. Thanks so much, Simon, for the fantastic presentation. And now I'm very pleased to introduce our next presenter, Professor Michael Goard. Michael, thanks so much for joining us today, and the floor is yours whenever you're ready.
All right, so thank you, Liam, and thank you to NeuroTAR and to Insight Scientific for hosting the webinar. So today I'm going to tell you about some recent work from our lab, this is all unpublished, on cellular resolution imaging of the transverse hippocampal plane in behaving mice. So now, those of you who know me or my work at all may be wondering who let this visual cortex guy into a talk about the hippocampus, and that's a very reasonable question. And the reason kind of how we found ourselves here, somewhat by accident, is that we've become very interested in what mouse vision really accomplishes for the animal. So when we talk about primates, including humans, we use our vision for recognizing that conspecific across the street or seeing that ripe piece of fruit hanging from a tree. And mice tend to use their nose for such functions. And so that led us to start asking, what do they use vision for? They still have a considerable portion of their brain dedicated to visual processing. And one thing that the nose doesn't really do for the mice is allow them to understand their spatial environment. If you want to know where some obstacles or boundaries are, or where a potential predator or prey is located, their vision is very helpful. And so we've become really interested in this idea that the mouse visual system gives kind of process inputs to parts of the brain that are involved in spatial navigation. So this could be useful for determining place cells, for example, in the hippocampus, for head direction, for kind of aligning our orientation relative to the environment in the retrosplenial cortex and other areas, as well as our understanding of where boundaries are in the environment. So this is an egocentric border cell at the bottom here, which were recently discovered, where we kind of know where obstacles are relative to us. And all of these are going to rely heavily on visual input. So this is how we became kind of interested in this topic. And then we ended up finding ourselves kind of curious about recording from some subcortical activity. And we realized that some of the applications that we developed for recording cortical activity might actually be helpful in this way. So I want to tell you a little bit about this, and this is very new work from the lab, so it's still a little bit raw, but I think some of you might find this interesting for your own research. So I'll just go through this briefly since Simon already talked about it, but there are some common approaches to measuring hippocampal activity. I'm sure you know about all of them already, such as the tetras, where you have a microdrive cemented to the skull, and then you can independently raise or lower bundles of microwires to record spike level resolution neural activity. This is really nice if you really want to know exactly when the spike times are happening. It also allows you to record from freely moving mice, but there are some disadvantages as well, such as it can be difficult to spatially localize which cells you're recording from. You can't really use genetic identifying markers as well. And some of these are ameliorated by two-photon imaging approaches, such as shown on the right here. So here you actually remove some of the cortex and place a window on the hippocampus. And the advantage here is you get much higher spatial resolution. You can label cells with fluorescent markers. You can image the same cells over time. You can even do subcellular imaging. The disadvantage, of course, is that the mice need to be head fixed. You have much poorer temporal resolution, and there's some damage caused just by the prep. So, again, both of these are very useful, and we've learned a lot by doing these types of recordings. Now, we decided to develop a different sort of recording modality using two-photon imaging. So it has some of the same disadvantages as the traditional two-photon imaging from the top. But what we wanted to do is something that neither of these approaches really get at, which is being able to image the entire transverse hippocampal circuit. And so it's been known for many years that the hippocampus has this really beautifully extrapolated circuit. So this drawing was done by Santiago Ramon y Cajal. I'm guessing many of you have seen it before, over a century ago. And just using the Golgi stain, he was able to infer the connectivity within the circuit. such as the input arriving in the dentate gyrus and being sent to the CA3 and then to the CA1 and eventually out of the hippocampus and rhinal cortex. There's some other pathways as well, but it's kind of the primary one. And we think that there's a lot of action happening within this circuit because as stereotype is repeated all down the elongated hippocampal structure. So we really wanted to be able to try to image this circuit and ideally the whole thing simultaneously using light microscopy. And so this is a project that has been fronted by Will Redman and Nora Wolcott, two graduate students in the lab. And I've actually also been involved in some of the surgical approaches, so it's been a fun project for me as well. And the idea here is that we wanted to use microprisms to be able to see down deep into the hippocampal structure. So for those of you not familiar with this approach, it's been used in the past for imaging from cortex and other structures. And the idea is that you have this glass prism which passes the light, and then you have a reflective surface at a 45-degree angle along the bottom. In this case, it's coated with aluminum, which reflects the imaging plane 90 degrees. So rather than imaging in parallel with the surface of the cortex, you are imaging in the transverse direction. And what this allows us to do is, so first, let me give you an example of how we can use this in visual cortex. So this is recording from the lab. And here you can actually see all the different layers of the cortex. So this is layer 5 down at the bottom. And you can see the layer 2, 3 pyramidal cells up at the top. And then in the middle, if you look closely, you can see some of the smaller stellate cells blinking in layer 4. And so we can use this in cortex, and this was originally developed by Michael Levine's lab. But what we wanted to do is see if we could actually extend this down to the hippocampus. And so the idea here is that we're using kind of a longer prism, and it includes kind of essentially you can think of it as the tube on a periscope that allows us to get all the way down through the cortex into the hippocampus and then reflect the imaging plane 90 degrees so that we get that nice transverse cross-section through the hippocampus. And so the idea here, so taking a sagittal section, this is from the Paxos and Franklin Mouse Brain Atlas, you end up seeing a cross-section through the dorsomedial tip of the hippocampus. And it's about 1.5 millimeters on the side. And so it's shown here with the red square on the bottom figure. We do have a smaller prism design. So if you're only interested in looking at V1, you don't need to if you're only interested in seeing CA1 and you're not interested in seeing the rest of the hippocampus, you can use that smaller prism and it causes less damage. It's a little easier to do the surgery. Okay, so let me cut right here to kind of the pretty pictures right away. So this is actually taken using this prism imaging approach from an awake, behaving mouse, and this is a Thigh1 GFP-M mouse. Some of you may be familiar with this line. It was one of the first lines used for subcellular imaging in the early days of two-photon imaging. And what you can see is it sparsely expresses GFP in a subset of the excitatory neurons. Now, for some reason, the dentate gyrus is actually a much more dense expression. In the CA1 and CA3, you can see this is the cell body layer going along the top and around the left side, and there's just a small number of neurons. And when you zoom in, as you can see on the right, you can see the elaborated dendritic structure of those neurons. And then the dentate gyrus, as I mentioned, is much more densely expressing the indicator. So this just shows that the technique works. Again, this is an intact mouse. They don't know we can, image their hippocampus while while they're running around, for example. And so they do need to be head fixed, but they can, as I'll show you later, they can be running around on a moving platform. And so you may be wondering, probably the first question that came up to us is how much damage are we doing? And the answer is we are doing some damage, but it's not totally extensive. So what I'm showing here is some histology we did where we looked at the lesion site and then stained for GFAP, a marker of astrocytes. We've also looked at microglia and some other markers to look for potential cellular damage. And there is slightly higher expression of GFAP and microglia right on the surface of the prism, but it kind of goes back down to kind of basal levels very quickly as you go away from the prism. It doesn't seem like we are causing kind of massive damage throughout the whole hemisphere. It's more localized to just in front of the prism face. I should note here that the lesion site you see here is much bigger than it actually is. It's because, when you do the histology, the tissue kind of shrinks away from the lesion site a bit. As kind of further confirmation, we just want to make sure we can continue to image kind of the same structures over time. And so these are actually two images taken over 100 days apart from the CA3 of the hippocampus. This is using a different transgenic line for GCamp, which I'll talk about later. And you can see the same cells are replicated, over 100 days apart. You can even see some of the same blood vessels. There's a tiny bit of change, but you can see that the structures are not, changing massively. We're not getting massive reorganization or anything like that. So this made us hopeful that, we can actually do some kind of quality science using this approach. Now, next, we wanted to characterize and make sure we're not losing a lot of resolution with this approach. The whole advantage of two-photon imaging is that you get really nice subcellular resolution. And so we image these fluorescent microspheres. They're smaller than a micron. So the idea is that we can use these in order to see, how big it appears on our image when we use our two-photon imaging. So first we did it just as kind of a control. We imaged these microspheres through a standard cranial window, and then we did the same thing through the hippocampal microprism. And what you can see is that so this is the XY profile of the bead, and you can see it's pretty focused at that particular spot. And when we look at the profile on a linear axis, what we see is that the diameter of this submicron bead appears about 1 micron on our image, which is pretty good. It's actually close to the diffraction limit of light. So this is about as good as we could expect given the numerical aperture of the objective we're using, the wavelength, etc. And we get similar values when we do it through the prism. So you can see it's a little blurrier here, but when we look at the diameter of our apparent bead, it goes from 1 micron to 1.2. So we're not complaining too much about that. It's a little worse in the axial direction. So this is going through the cranial window, it'd be in the Z plane. Going through the prism is actually in the Z plane away from the prism. Here we see a little more spread, so it's about 4 microns through the cranial window, and it goes up to about 7 or 8 microns through the prism. So we do have slightly worse resolution through the prism, particularly in the axial direction. However, this is not terrible. I mean, this is still less than the diameter of a cell body, so we don't think it's going to cause major issues with our two-photon imaging. All right, so if this is true, then we should actually be able to image subcellular structures in the hippocampus. So next we tried to do that. And here, what I'm showing you is a zoom in on some CA1 neurons, and we've kind of zoomed in on a particular dendrite. Maybe a little hard to see, but if you squint, you can actually see some of the spines dotting the dendrite. They are a little bit harder to resolve than when we image spines in cortex, but using some image processing, we're able to pick them out, and we can actually see the same spines across sessions. So we know that we're not, we're not just kind of over filtering noise or anything like that. We've done some other checks too, using the high resolution images from cortex. And we're able to kind of recover the structure of the spines over time. And we can even differentiate them into different spine classes. So these are kind of the averages of the different spines we're able to differentiate, including the filopodium, the mushroom spines, the stubby spines, and the thin spines. So for these, what we did is we basically took all of the ones that were identified a certain way and averaged them together. So you can see the profile here. And they were discriminated using, various aspects of their size and ratios of their head to neck diameter and so on. But this allowed us to do is actually track the same dendrite over time to see new spines being added, spines going away. And so we see fairly typical distributions of spines. So we see very few filopodia, which is typical in adult animals. And then the rest are kind of divided between thin, stubby, and mushroom spines. And in terms of stability, the filopodia are the most unstable. They turn over quite quickly, as has been shown before, while our mushroom spines are the most stable. So all of this is kind of in accord with what we expected to see. So can we actually do functional imaging with our microprism approach? So here what we're going to do is actually use genetically encoded calcium indicators. So these are mice that express GCamp6S in excitatory neurons. We're using a glutamatergic transporter to drive expression. And we're zooming in here on CA1, and you can see a number of these CA1 neurons kind of blinking. This is actually just during spontaneous activity, but you can see quite a bit of activity, and sometimes you can actually see the dendrites kind of lighting up, presumably from backpropagating action potentials. And we can also see activity in the dentate gyrus. So this is looking at kind of both parts of the dentate gyrus, the more dorsal and the more ventral. And the activity in dentate gyrus is a bit more sparse, especially during spontaneous activity. But we can resolve bouts of activity in these neurons as well. So just to give you kind of a better idea of what we're seeing here, I wanted to do a little bit of a guided tour through the prism as we're imaging. So we're actually going to start in the cortex. So this is the cortex just right in front of the prism, just to show you that the cortical neurons seem to be behaving normally. And now what you're seeing is actually the edge of the prism towards the bottom of the screen, that kind of sharp line. And now we're going to translate over to the middle of the prism, and then we're going to rapidly go down in the z-plane through the prism until we can start to see the hippocampus. And so now you can see the CA1 and CA3 neurons lighting up. And then we're going to zoom in and look at our specific regions. So we're going to translate over to CA1 first, and so you can see some of the neurons there. This, again, is just spontaneous activity. Later I'll show you some activity while the mice are moving around. Now we're going to move over to area CA3. And then finally, we're going to translate over to dentate gyrus. In this particular mouse, the dentate gyrus was a little bit out of plane, so we have to change our focal plane a bit. And then we're going to zoom in so we can see those smaller granule cells in the dentate gyrus, and you can see a lot of activity in them as well. So just to kind of give you an idea of what is happening as you kind of pan around within this microprism. Yeah, one thing I forgot to mention is the way we actually implant it is we take a diamond microblade and use a manipulator to lower it, going through the dura and through the cortex into the hippocampus, and then slowly lower the implant, again using a manipulator, into that incision. And so what that does is it kind of pushes the tissue out. And so the cortex kind of around the incision site, it all seems to do just fine. We can find activity in those neurons even months after the implantation. We are cutting connections, of course, so it's not like we're not doing any perturbation, but there's less damage than, say, if we had to aspirate out the cortex overlying the hippocampus. Okay, so now how do we actually measure spatial responses in these neurons? So here we use the neuroterminal home cage system, and the idea is that the mice are moving around within this chamber. We can put visual cues along the side. We actually use this kind of nylon paper, so it's relatively water resistant. And so we can have these nice cues that the mice can use to orient themselves. And then we use the tracking system in order to record the location of the mice as they're moving about. So we can record place fields using this approach. And so we did it for a while in the open field. We had a little bit of trouble because sometimes they wouldn't fully explore the open field environment. They'd stick to the edges or kind of hang out in one area, not another. And so we actually, from advice from a postdoc in Simon's lab, we decided to move to a circular track where we found that they repeatedly, as they circle around, they kind of repeatedly sampled the environment. And as a result, we were able to get kind of more robust place cell activity. And so that's what I'm going to show you from here on out. So first, I wanted to just kind of quickly give you an overview of some of the spatial activity we saw in each of the three major subregions, CA1, CA3, and dentate gyrus. So first in CA1, here we have a kind of typical field. This is a maximum intensity projection. So you can see there's quite a few neurons and even the dendrites. And we found that we can actually measure place fields even in the dendrites, which is kind of cool. And then these are a number of example place fields. So you can see they're nice. They respond to particular positions in the track. That bottom one, left one, I should point out that the track, because it wraps around, that's not like a bimodal field. It's a single field that's just kind of wrapping around from 80 back to 0. We also saw a number of cells that were modulated by the speed of the mouse. And the other thing I want to mention about the place fields is that they span the track. So you can see that they span pretty evenly if you look across a number of the cells that we recorded. And we also found cells that are modulated by the mouse's speed. So these were modulated kind of monotonically. So some increased their activity with speed and some decreased. And then kind of summarizing the results, we found that about 20% of the neurons in CA1 had nice significant place field activity. 5% or so had significantly modulated speed responses. There was just a small percentage, 1% or so, that were modulated by both. And then roughly 74% were not responsive to either place or speed. Looking at CA3, we see somewhat similar results. Again, there's our example field. Again, nice place fields that span the track. We also see some speed cells in CA3. And looking at the summary, we actually see slightly fewer place fields in CA3, but relatively more speed-driven cells. So as a result, again, it's about 25% of the total cells are kind of modulated either by speed or by position using this approach. And then finally, we looked at the dentate gyrus. And again, we do see some nice place fields. We think they were maybe a little bit broader, though this is not statistically significant. We have fewer cells in the dentate gyrus. It's just a bit harder to record from there, and they're kind of a little less active. But you can see that they tend to span the circular track, and we also find speed-modulated cells. And here, again, we actually find an even smaller percentage of cells that are significant place cells, but still 13% or so, and then a similar number of speed cells as to CA3. So so far I've showed you that we can do both structural and functional imaging throughout the subregions. So I wanted to go through some of the advantages and disadvantages of the approach, and I don't want to kind of oversell the approach. I'm not advocating that everyone throw away their tetrodes and dump their two-photon prep into garbage. There are some disadvantages of this approach. So the implant does cause some damage to the cortex, probably a little less than the traditional top-down two-photon imaging approaches. But one important thing is that it also causes some damage to the dorsal medial tip of the hippocampus, which is the actual structure we're imaging from. So that needs to be kind of kept in mind. It's an important caveat. We try to go as medial as possible, so we're cutting into as little bit as possible. However, there might be connections that are severed, between that part of the hippocampus we're imaging from and other regions. Another disadvantage is this surgery is non-trivial. It definitely requires an experienced surgeon. It took us a while to master it. Once you get good at it, we have a pretty high success rate at this point, though it's not 100%. But there's certainly kind of a training step involved. The spatial resolution, as I mentioned, is a little bit lower than imaging through a window, though it's not terrible by any means. And then finally, we do have a lower yield compared to when you're imaging parallel to the cell body layer. So when you're doing the top-down CA1 recordings, because your whole field is filled with cells, you can get many more at a time. For our recording approach, you have these bands of cells, and there's a lot of cells in those bands, but we're typically talking, between 1 and 200 cells at a time, which is a little bit less than the top-down approach. Though I still think that's pretty nice, coming from an electrophysiology background. Okay, so some of the advantages of this approach is it allows you to see all of the subregions. You're not limited to just CA1. And, I know people have tried imaging deeper using other approaches, but it often requires kind of compressing the hippocampus in order to get there. So this really allows us to see all the different regions, and we can even see them simultaneously. So unfortunately, I don't have any videos of this, but recently we made some modifications to our microscope so we can really image the entire hippocampus all at once. And you can see CA1 and CA3 and dentate gyrus and all the neurons lighting up at various points. And so I think this could be really interesting for studies where you're looking at interactions between regions as they're doing spatial coding or memory tasks, for example. Another advantage is that we can identify different neural subtypes. So some of this is using the typical approaches, labeling them with a red fluorophore, for example. But we can also see some cell types which might be difficult using traditional imaging approaches. So, for example, we can it would be much easier to differentiate CA2 cells as opposed to CA1 or CA3. We can zoom in on the mossy cells and the dentate gyrus. So these cell types that are typically kind of hard to find using other approaches are much more readily identifiable. We can, I didn't show any data, but we can do chronic imaging of the neurons across sessions. We found, as Simon did, that they tend to remap quite quickly. But certainly within sessions, we can look at stable encoding across environments, which is pretty cool. And then finally, and I think this is actually one of the biggest advantages of this approach, is that because of the imaging plane, we can see the entire dendritic arbor of the CA1 and CA3 neurons. So you can imagine there's some really cool experiments where if you more sparsely expressed a GCamp, you could actually zoom in on individual spines or segments of dendrites and see how subcellular place fields contribute to the ultimate place field of the neuron you're imaging. So for people interested in dendritic integration or subcellular imaging, I think there's a lot of cool stuff to be done here. And then finally, I wanted to just leave you with an example use case of how this can be used for doing some interesting science that you couldn't do with any of the other approaches. So the one I wanted to do here is a very new direction. So in the past, I've been interested in neuromodulation. So we're thinking of looking at the effect of neuromodulation on the hippocampus. And then it turns out my wife is actually a neuroendocrinologist, and she said, why don't you look at the effect of hormones on the hippocampus? Because it's been known for some time that throughout the estrus cycle of the mice, you actually see differing spine densities. So this is a classic result from Catherine Woolley and Bruce McEwen's lab, in which they looked at CA1 apical dendrites, and they found an enrichment of spines during the proestrus stage and a reduction during the estrus stage, with kind of more intermediate numbers during the other stages. And so the first thing we want to do is just see if we can actually see this in vivo. Can we actually see spines kind of growing and shrinking in female mice as they go through the estrus cycle? And so what we did is we started with our mice, and then every 12 hours for 8 days, so Nora, who did these experiments, had to do some fairly heroic experiments where she would use vaginal lavage to take samples of the cells in order to do cytology. And then we would later use this for identifying what stage of the estrus cycle the mice were in. And then she would also image spines within the CA1 apical dendrites. Oh, I should mention that this effect that Woolley and McEwen found was located in the apical dendrites specifically. So it would be hard to do with some of the other techniques, which might let you get to the basal dendrites on top, but nothing else. So then what Nora did was take these images and run them through a machine learning network that she developed for identifying the stage. I won't get into that here, but it's kind of a nice way of doing it if any of you are interested. And then lined it up later with images of the spine. So after we had kind of annotated the spines and figured out which ones were added and subtracted, we lined them up with the specific estrus stages. And the experiments were all done, and all the analysis was done kind of blind to the stage of the mouse to make sure we didn't kind of bias our interpretation of the data. But what we found is that sure enough, we saw kind of starting with estrus here in this leftmost picture of the spine, and then we imaged the same spine for 8 days. And we found that as they went through the cycle, so here I'm just showing 4 days, as they went from estrus to proestrus, we tended to get net addition of spines. There were some spines that went away as well, but there was kind of a net addition of spines, resulting in a higher spine density during the proestrus cycle. So this is shown over 2 cycles here. And it looks like a subtle effect, but maybe if you compare proestrus, which is so that kind of second bar and then the 6th bar, you see there's about 10% more spines than average. And if you look at estrus, which is the 3rd and the 7th bars, you'll see there's about 10% less spines than the average. So there's a kind of a net change of about 15 to 20% just over the 24 hours from proestrus to estrus. And it's hard to imagine that a 20% change in spine density is not going to have any functional effects. So what we're really interested in looking at next, so we showed that the spine density is highest in proestrus and lower in estrus. What we really want to look at next is what functional consequences does this have? Does it affect, place field remapping, anything like that? So, there's kind of an exciting next set of experiments to do. We're also doing much more characterization of, which spines are appearing during proestrus. Are they stable? Do they stick around? Or are those the same ones that disappear during the next cycle? We're really interested to see what's going on there. So hopefully to serve as kind of a good example of how this approach can be used to do some interesting science. And as you can imagine, there's all sorts of other things that I think would be really interesting to look at. You can look at the effect of neuromodulators. You could look at altered protein expression. So a number of you might be interested in disease models, Alzheimer's models, things like that. I think this approach could be really useful for looking at both structural and functional changes with those models. pharmacological agents, etc. So I think there's some cool science to be done, while keeping in mind the various caveats of this system. All right, so that brings an end to my part of the talk. I wanted to thank you all for listening. I also wanted to give special thanks to my lab and for this project, especially Will and Nora, who led the project. As I mentioned in the beginning, most of my lab is actually focused on other projects looking at how visual input influences retrosplenial spatial responses, and we're using some of these systems for doing that work as well. So if you're interested, get in touch. If you ever want to come visit UCSB, it's not a bad place, so drop a line. And so finally, I'm going to turn it back over to Liam.
All right, fantastic. Thanks so much, Michael, for sharing your research with us today. And now we are going to move on to the Q&A session. And I'm just going to add Professor Goard and Professor Schultz to the audio line here. Okay, all set, Simon and Michael?
Yes.
Yep.
Perfect. Okay, let's jump right in with a great question for Simon. How does head fixation affect spatial memory?
Okay, I mean, that's an interesting point, which was forefront in our minds when we started this project. Clearly, our mice are head fixed, and because they're head fixed, they don't have vestibular information, or more to the point, there's sort of vestibular asynchrony with respect to the information you would expect them to get as an animal is sort of naturally moving around its environment. So we certainly sort of went into this expecting upfront actually not to see place cells in 2D as a result of that. We knew, however, that from virtual reality studies that we should perhaps see place cells in one dimension in sort of tasks where they're sort of, say, traveling through a track like our circular track task. And we did see that. But surprisingly, we also saw them in 2D. And, really our thoughts on that were that, well, certainly vestibular information is important, but actually maybe it's not the full story. And maybe a lot of the issue here is actually multimodal information, that we have tactile information, we have visual information, possibly we have olfactory information, etc. as well. And actually, if the animal is able to integrate this all in, maybe that overcomes the lack of vestibular information. So actually, so it does affect it, I think, but not as much as we expected. And we were able to map place cells even in two-dimensional environments, which indicates that spatial memory is there.
Perfect, great answer. Michael, I'll ask you to lead this next question, but do other types of cues, for example, tactile cues on the floor, improve place field measurements?
Right, so we might need to ask Simon to come on this because I think he actually looked at it a bit more than I did. But so we did use tactile cues, and we did it less so that the animals could kind of tell where they were within the environment, but more as a way to kind of help them clock how far they had gone. So, for example, in the circular maze, I guess you call it, or track, we had regular kind of cues just so that if they were running fast, they could kind of have some indication of how fast they're going from the tactile input. As Simon mentioned, they are missing their vestibular input, so we wanted to kind of give them some extra information. However, we used the same cues, so we didn't try to distinguish location based on the identity of the cue. And I'm not sure how much that would help because, remember, they're head fixed in place, so it's not like they can really get down there and whisk and help kind of distinguish different objects. They're really only kind of doing it with their paws. And I'm not sure, without kind of spending some time training them, if they could distinguish subtle differences in cues just as they're moving about. So, in terms of how much it helps, I actually don't have a great sense. Simon might have some insight on that.
Sure. Yeah, Simon, do you have anything to add on to that?
Yeah, I mean, I guess I would just add that, we had both visual and tactile cues for similar reason. We just wanted to create the richest local environment possible. But what we didn't do is try and, dissociate those two things. And that would actually be a very interesting experiment to do. It's probably hard to do it properly because, of course, okay, you say they're not necessarily sort of getting down and whisking, but of course they do have whiskers, and if they go through a narrow corridor, maybe they can feel our visual cue, etc. Maybe they can see our tactile cues. Hard to know. So, we don't really know which is the most important there, to be fair.
Excellent. Next question here. Yeah, how long did it take you to train mice in the airlifted environment? Michael, maybe you could start with this one.
Right. So what we ended up doing, and again, part of this was just kind of getting them used to the setup and making sure they were running enough, so we didn't really thoroughly empirically test it. But we ended up first putting them in the chamber. So we did this over 2 sessions over 2 days, and then we put the whole chamber in a larger box so that they could just explore it while they're freely moving. And that just kind of let them learn the local cues and get comfortable in the environment. And then we spent about 1 week of daily head fixation sessions where they would run around on the mobile cage system. And we found that usually the first day they were a little bit scared and wouldn't run around a whole lot. There was some individual variability between mice. And then over that week, they would creep up a little bit. Typically, they're running at about 20 or 30% of the imaging session. I suspect if we kept going even longer, we could raise that even a bit more. We've generally found with running wheels and things like that, they like to run around. So once you get them really comfortable, they might spend more time on there. But we just, after a week, we kind of felt like we were hitting a plateau. So we stopped training them at that point.
Excellent. And Simon, what was your experience like with habituation of the animals?
Yeah, similar, about a week. It depends on the particular sort of environment. It took a little longer to get them sort of nicely running around the open field than the 1D task. We did water restrict. Okay, so our animals were rewarded effectively on the basis of how much they ran around. Okay, we wanted to get them exploring as much so that we could map place fields well. After a week or so, we were up to 50 to 70% of time exploring or time moving in the environment, which is as much as you want, really. You don't want them doing it all the time.
Yes, that's a pretty large difference, and I think that's to do with how motivated they are based on the water restriction.
Yeah, exactly.
Perfect. Simon, I'll give you this next question here. Don't you need distal visual cues to form place fields?
Well, that's kind of the received wisdom in the place field community, but apparently not. I should note that the mouse visual system is actually adapted to see things close up. They don't have particularly high spatial acuity. If you think about it, features far away have to be big for them to be able to see them. Features closer up can be a little smaller and they can see them. It's worth noting that, in our environment, within the two-photon microscope cage enclosure, it's dark. They can't see any distal cues because, that would be a confound because of the way the environment is set up. So it is only local cues and yet we find place fields. So you do get place fields from the local cues only. And there are previous papers that support that also.
Okay, interesting. And Michael, I'll get you to lead this next question. When imaging through the microprism, is there a significant tissue movement relative to the prism?
Right. So it's actually not too bad. So you also get some movement, but we actually find there's less movement when we're imaging through the prism compared to when we're imaging through a window, for example. So now I haven't imaged hippocampus through a window, so I can't really compare, but I know imaging cortex through a window, we see a little bit of movement, both lateral and Z movement we try to avoid, but especially when they drink, you get quite a bit of Z movement. With the prisms, both in cortex and in hippocampus, we do not see a whole lot, and we think it's because there's kind of a second anchoring point that keeps the tissue stable. So it's actually quite nice for dendritic imaging. I imagine if you were doing axonal imaging, it would be nice for that as well.
Excellent. Next question here for Simon. You said that you see slightly faster remapping of place fields compared to in freely moving mice. And so why do you think that is?
We honestly don't know. The same applies to the virtual reality papers if you look at them carefully. So somehow there is something about freely moving behavior which sort of stabilizes place fields and therefore presumably sort of stabilizes spatial memory on a slightly longer time course than you get with head fix preparations, including virtual reality and including the mobile home cage type environment, floating cages. So that's interesting. I think it would be interesting to try and pin that down at a later point.
Yeah, definitely. Michael, I'll give you this next question. Do you think this imaging approach could be used during other behavioral tasks? How do you kind of see that playing out?
Sure. So with the microprism imaging, we could use it for all the same types of imaging circumstances that we'd use for other imaging approaches. So, for example, with the mobile cage, there's been some attempt to build these T-mazes where you can train the mice to alternate for a reward. You can do all the same virtual reality tasks that people typically do. So pretty much any head fix task you could run with this approach. One interesting thing I didn't really talk about during the seminar is, it's well known at this point that there are cells in the hippocampus that encode durations of time, the time cells. And that would be a very interesting thing to look at using other sorts of tasks.
Definitely. And I think in the interest of time, we'll make this next question the last one. And I'll get both of you to respond to this one, but maybe Simon, I'll get you to answer it first. But why did you choose the mobile home cage over other approaches, like you said, a treadmill and VR? And what do you think the advantages or disadvantages are in this case?
So, we had actually done virtual reality tasks before. We developed a platform in my lab previously for a closed loop treadmill plus virtual reality system for studying the cerebellum, in fact. But I guess my concern when we were moving to do a project on the hippocampus and on spatial memory was I wasn't really convinced that in the virtual reality environments the animals really know what they're seeing, in a sense. Clearly that if you're looking at, say, the early visual system, that's going to be fine. But we wanted to be able to do more complex things like look at two-dimensional place field environments, etc. And I thought our chances of getting that working nicely were best the more sort of permanent, if you like, the information could be. And I would really say that's the one advantage of the mobile home cage system, in that there is a sort of a permanence of the environment that the animal is perceiving. That's a plus and a drawback as well. Obviously, with the virtual reality environment, you can reprogram it on a rapid basis, and there are some questions you can ask with that you couldn't do in another way. But nevertheless, the physical environment, permanent physical environment, is what the mouse's sensory systems and cognitive systems evolved to deal with, and it's what they're adapted to. So therefore, I think it has particular strength for looking at cognitive tasks, in fact.
Excellent. And Michael, why did you choose the mobile home cage?
Right. So I think I'm pretty much echoing Simon. So, we've also done some virtual reality in lab, and what we find is I think it's great if you want to be able to control the task. I mean, you're kind of unlimited in terms of which task designs you can use. You can switch environments, you can warp them around. So it's great for that flexibility. However, I think for studying kind of spatial representations in the hippocampus or other areas, there's something about having a real object. When they run into a wall, they bump their nose on it, they can whisk against it. When they run into objects, the same thing. So there's kind of more meaning to their local environment rather than when they're in VR and they bump into a wall. They're really just the visual stimulus is no longer updating. There's no real meaning to it. So I think for kind of promoting spatial coding, the floating cage has some real advantages. Now, neither of them you get the vestibular input, so that's something I think as a field we still want to work on and try to come up with even more realistic solutions where we can image while we get kind of all the inputs that you're getting as you move around in the world. But I think this is a step in the right direction.
Definitely. The research is already so fascinating. I can't wait to see where it goes in the future. And yeah, so thanks so much, Michael and Simon, for sharing your work with us today. It's been a real pleasure having you with us.
Thank you, Liam.
Thank you.
Yeah, and a big thanks also to the audience for joining us today. And a big thanks to Neurotar as well for helping to make this event possible. So thanks again for taking part in this Inside Scientific webinar, and we'll see you again next time. Have a wonderful day, everyone.